Okay, so last class you would have completed a web quest on the different types of stem cells and their functions. Today and for the next class, you're going to be working on 5 scenarios and determining what the advantages, disadvantages, and which type of stem cell would be best for each type of disease. So they give you a little scenario at the top, and then you're given a table. Now, I am not putting this into Kami. I'm using it in Kami right now for the video, but for you guys, I want you to make your own Google Doc or even just enter it in the text box just in case Kami doesn't work correctly. So you guys will have to have a separate document. And I had you do that web quest to get you used to kind of looking up what these different stem cells do, and that way it's kind of fresh in your memory so that you may need to do a little more additional research, but you'll know the direction that you're going in. So I'm going to walk you through the first scenario, and then I'll let you guys kind of have at it on your own here. So type 1 diabetes results when cells in the pancreas don't produce insulin. Without insulin, sugar builds up to toxic levels in the bloodstream, damaging the kidneys, eyes, heart, and nerves. Patients manage their diabetes by taking insulin, regulating their diet, and closely monitoring their blood sugar levels. However, the constant nature of diabetes management can be overwhelming, and it only addresses the symptoms of the diabetes. This is not a cure. So you're a researcher who would like to develop a cure for diabetes. You want to engineer insulin-producing cells from stem cells and transplant them into diabetes patients. Special considerations: the stem cells should be easy to obtain and manipulate in the lab. The stem cells should be able to differentiate into pancreatic beta cells that produce insulin. The stem cells should not trigger an immune response in the patient. So these are things that you're going to want to really pay attention to, and that'll help you to list your disadvantages. Which type of stem cell should you use? In the table below, list advantages and disadvantages of using each stem cell to treat type 1 diabetes. Circle the stem cell type you think would be best. Instead of circling it at the end of your table, you can just tell me which one you think would be best since you're typing this out. And then explain why you think the cells you chose would be best. So you might want to go in and do some research, even go back to that website that you were on yesterday. And that will help you out as well. So let me tell you a little bit about the information that you should be looking for and what you'll be finding. So researchers are working to develop a stem cell therapy, and that's going to regenerate insulin-producing pancreatic cells in type 1 diabetes patients. Pancreatic cells are lost in these patients when their immune system attacks them, which is why it's important that you don't trigger an immune response in the patient. A recent clinical trial involving 23 type 1 diabetes patients showed that 20 became insulin independent for at least 4 years after homeopoietic stem cell treatment. Homeopoietic stem cells give rise to blood and immune cells, and the goal of this type of therapy is to reset the immune system and improve tolerance of self cells in patients. So that would be like your somatic cells, because from our web quest yesterday, we know that the somatic or adult cells, they are going to produce blood cells. And your blood cells have a lot to do with your immune responses. Next, we'll talk about the pluripotent and the embryonic stem cells, the ES stem cells. So you have other researchers that are producing a different strategy. They can be, these pluripotent or the embryonic cells can be directed to differentiate into functional pancreatic beta cells in a dish. Researchers at the Nevada Cancer Institute transplanted iPS cell-derived beta cells into a diabetic mice to restore insulin production and normal glucose levels. This experiment has not been carried out in humans. However, islet cell transplants from cadavers serve as a proof of concept that the cell therapy can successfully restore control of blood glucose levels in patients. Now, a third approach to curing diabetes would be to activate adult stem cells already present in the pancreas and direct them to become beta cells, which is kind of what we were looking for. Researchers have successfully used this approach to restore pancreatic function in mouse models. And similarly, researchers have been able to obtain certain types of differentiated pancreas cells that would normally produce digestive enzymes into insulin-producing beta cells. So right off the bat here with the somatic cells, from that web quest yesterday, you know that these are difficult to obtain. So that would be a big disadvantage. When we talk about the IPS ones, again, from what you did yesterday, you know that this particular type of stem cell is a big safety concern for the patient because they haven't been tested in humans. When you have embryonic stem cells, this can trigger an immune response. And with cloning, it's time-consuming, it can be inefficient, and it's also expensive. Now, if we want to look at advantages for these embryonic stem cells, we would say that you can direct them differentiate into pancreas cells. And that would be the same for this one down here, the PS cells. For your cloning of the cells, an advantage would be that this has been done. In mice already, and you can make them into any cell. And for my somatic cells, you'd be resetting the immune system. Now, if I looked at those advantages and disadvantages that I have listed there, I'm going to take into consideration my special considerations box up here at the top. If we were looking for something, let's say it needs to be easy to obtain, that would take out my somatic cells because they are very difficult to isolate and grow. We also, down here, we don't want to trigger an immune response, which we know embryonic stem cells will trigger an immune response. And it says the stem cells should be able to differentiate into pancreatic beta cells. I would probably go with the PS cells or even the therapeutic cloning cells because you can direct them to differentiate into either one of those cells. So I would circle one of those two, and then I would try to back that up with why I think that would be the best cell. So your answers may vary from other people's in the class, and that's okay as long as you can back it up to me. So some of your advantages and disadvantages will be very similar as you're moving through these different scenarios. Let's go ahead and look at number 2 together as well, and then I'll leave you guys on your own for doing heart disease. This one, stem cell characteristics, and lupus. So Parkinson's disease is characterized by the gradual death of nerve cells in the center of the brain. As the cells die, they can no longer send out signals that coordinate basic functions such as movement, attention, and learning. Therapy for the disease focuses on medications that mimic the chemical messengers the dying cells should be sending out. The drug therapy mainly slows the course of the disease, but is not a cure. You're a researcher who thinks it may be possible to devise a replacement stem cell therapy for Parkinson's disease. Your aim should be to create nerve cells from stem cells, then transplant them to the midbrain, where they will carry out normal function. So for which type of stem cell should you use, we're going to be doing the same thing we did in the last one, listing advantages, disadvantages, and then saying which one we would do. Special considerations: the stem cell should be easy to obtain and manipulate in the lab. It should be able to differentiate into neurons that produce the proper chemical messenger. The stem cells should not trigger an immune response in the patient. So let me tell you a little bit about Parkinson's disease then, and the different types of stem cells that they have available. Parkinson's disease is a prime candidate for stem cell therapy. This progressive neurological disease is caused by the loss of midbrain neurons that produce the neurotransmitter dopamine. Dopamine coordinates movement, learning, and the reward pathway. Stem cell therapies have been attempted in clinical trials for Parkinson's disease since the 1980s. Most of these experiments used dopamine-producing cells obtained from fetuses. Although the transplanted cells survived and some patients benefited from the treatment, others suffered from severe lack of muscle control movement. As an alternative to fetal tissue, researchers are turning to pluripotent stem cells, both ES and iPS cells, as a treatment for Parkinson's disease. Researchers have already worked out the steps necessary to differentiate pluripotent stem cells into dopamine-producing brain cells. Recent work has been focusing on improving the safety of stem cell therapies. iPS cells are attractive because they eliminate the risk of an immune rejection, and a cell sorting technique separates undifferentiated stem cells, which tend to form tumors, from neurons before they are transplanted into patients. Transplanting dopamogenic neurons derived from either ES or iPS improves motor control in rat models of Parkinson's disease. A similar iPS cell therapy has not yet been tested in humans. So for looking for our advantages of embryonic stem cells, we know that they can become any cell. We know that that's also true for, I was wondering if I could copy that, for the iPS. Disadvantages: it can trigger an immune response. And here again, safety is a big risk. Because it's never been done in humans. Only in mice or rats. The somatic cells are difficult to grow and obtain. And for some reason, I can't type on here. There we go. And they can cause organ or tissue damage. An advantage to an adult stem cell. We know that they produce blood cells, which could potentially work their way up to the brain, I suppose. The therapeutic cloning. Wow. They're time-consuming and expensive. But an advantage would be that they have the potential to become any cell in the body. Now, personally, if I was looking at the special considerations in which cell I would want to pick, I would go with the iPS cells down here. Even though it is a safety concern and it hasn't yet been done in people, they're not going to trigger an immune response. They're not going to cause tumor growth, and they are fairly easy to obtain in a lab. So you guys will go through and do the heart disease one on your own. I will give you a little bit of information here about them, and that will help you to decide which type of cell you would want to use and go through and do your advantages and disadvantages. Just make sure you look at your special considerations as you're doing so. So just a little background information here for you. Since the first heart transplant took place in 1967, researchers have continued to develop new and improved treatments for heart disease. Stem cells have now emerged as a viable option for repairing damaged heart tissue. Bone marrow stem cells, for example, have been used in clinical trials to treat more than 1,000 patients after a heart attack. Researchers recently isolated adult stem cells, so that would be your somatic cells, from human heart via biopsy. They showed these cardiac stem cells can be expanded, then reintroduced into mouse hearts to regenerate the tissue. Similarly, cardiac stem cells can also be used to grow thin patches of heart tissue in a dish. Preliminary studies in rats suggest these tissue patches can be grafted into the heart to repair the damage. The long-term goal is to engineer an entire heart for transplantation from patients' own stem cells. This would eliminate the risk of immunological rejection. Human iPS cells can be led to differentiate into functional heart muscle cells and are therefore a good cell source for heart repair. Researchers at the University of Minnesota Center for Cardiovascular Repair have engineered a scaffold that will help them grow a heart. Starting with the cadaver's donor heart, they were able to remove all the living cells and leave behind only the extracellular matrix. The scaffold holds the shape of the heart and its blood vessels, allowing the flow of oxygen and nutrients as the heart grows. They plan to try both embryonic and adult stem cells to speed the heart scaffold. The hope is that the stem cells will migrate and differentiate to assemble a vascularized and fully functional heart. So right there, I'm telling you that you're either going to want to use ES or the somatic cells. It's now your job to determine which ones you think would be the best. We're going to do the other two scenarios, or you're going to do the other two scenarios tomorrow, so I'm going to stop there. Good luck.